Welcome to the Basic Science Plenary Talk. I'd like to start by acknowledging that we're gathered on Treaty 6 territory and homeland of the Métis, and I want to personally express my gratitude for the privilege of living on this land and my commitment to reconciliation. So I'm happy to introduce this year's Basic Science Plenary speaker, Dr. Guido Silvestri. Dr. Silvestri is the chief of the Division of Microbiology and Immunology at the Yerkes National Primate Research Center. In addition, he's the Georgia Research Alliance Eminent Scholar in Comparative Pathology and a professor and vice chair of research for the Department of Pathology and Lab Medicine at Emory University School of Medicine. He's also an editor of many journals, including Journal of Virology plus Pathogens, and has been involved in over 50 NIH studies sections. And I know he was involved in at least one CIHR VVP, which is where I first met Guido. Since 1993, Dr. Silvestri has been studying HIV pathogenesis, prevention, and therapy, mostly using non-human primate models. He's published seminal works on HIV immunology and pathogenesis, mostly using approaches that compare HIV with SIV pathogenesis, and recently published a paper that identified host genes that may explain why SIV-infected monkeys don't develop the AIDS disease. His current research focuses on using non-human primate models to study HIV persistence, reservoirs, and cure strategies, and has identified a role for CD8 lymphocytes in the control of SIV infections during ART therapy. So today he will tell us about new strategies for HIV eradication, lessons from Cd8 depletion studies. And join me in welcoming Dr. Silvestri.
Good morning, everyone. Thank you very much, Joyce and Linda and Kyle and the others for inviting me. It's a great pleasure and honor to be here and to be with many friends from the Canadian AIDS research community. One thing that Joyce didn't mention is that my real career started in Canada, in Montreal, in the years between 93 and 96, when I was a postdoctoral fellow with Rafik Sekali at the ERCM, and Eric remembers those years when our hair was dark and we were young and strong. And so my lab operations is connotated by a devastating lack of focus. So I do some work on AIDS vaccines, and we have a nice paper coming up next week that I will not talk about. We also do a lot of work on pathogenesis and comparative studies of monkeys that develop AIDS versus monkeys that don't develop AIDS upon SIV infection. And I will not talk about that line of work. But I thought that today I was going to talk about the work that at this point is more exciting, at least in my mind, and also more conducive to potential clinical applications. And I will talk about strategies for HIV eradication that are based on the definition of some new function of CD8 T cells that could be hopefully harnessed to achieve a functional cure for HIV infection. There is a tremendous amount of data suggesting that CD8 positive T cells are able to control replication of HIV and SIV. And a lot of the focus has been on the ability of CD8 positive T cells to kill productively infected cells that express HIV or SIV epitopes in the context of class 1 molecules, the so-called CTL activity. There is also reports of CD8 cells doing antiviral activity, playing antiviral roles through non-cytolytic mechanisms, including the production of antiviral cytokines or chemokines, and also through transcriptional suppression of virus replication. This is a line of work that was done many years ago by Professor Jay Levy at the University of California, San Francisco, that remained somewhat controversial because he predicated the presence of a CD8 antiviral factor, or CAF, a molecule that was unfortunately never identified, let alone characterized. So that work was eventually, received in the field with a certain degree of skepticism. As a monkey person, I always thought that the most convincing and compelling demonstration that CD8 T cells are really important in suppressing virus replication comes from the experiment where those cells are depleted in vivo in SIV-infected macaques. And those experiments are invariably indicating, and have been repeated by many different labs, that as soon as you remove the CD8 T cells from the system, you have a clear bump in virus replication that can go from 1 to 3 logs, even more sometimes. And so that there's a clear effect of removing these cells in enhancing virus replication. So the reason I got interested in this line of work, and the fact that has been really transformative, at least in my mind and in the mind of my colleagues working on this field, and hopefully when this body of work that I'm presenting today will all be published, it will be transformative for the field as well, comes from an experiment that I've done back in 2009. And it's an experiment that comes from a conversation that I have had in 2007 with someone who is in the audience today, Professor Mario Ostrowski from the University of Toronto. Mario and I had over dinner. Mario and I know each other since 25 years ago when we were together temporary migrants in Fauci's lab at the NIH. I was following Rafiq. He was there on his own. And the conversation was about trying to quantify the impact of CTLs on the lifespan of an infected cells. In other words, we know that HIV-infected cells, productive infected cells, from the famous model by Alan Perelson and George Shaw that was published over 20 years ago, we know that a productive infected cell with either HIV or SIV lives about a day and a half. And the question was, how long is it going to live, that cell, if CTLs are not in the system? If we remove the CTL, intuitively we thought that the cell should live longer, but how long will it live? Will it live, 3 days, 5 days, a week? And you can make that calculation by repeating the George Shaw experiment. So that's what we did, and that experiment was done separately by us, at Emory, and also by Satya Dandekar and Joe Wong at the California Primate Center. And surprisingly, we found that both in the early and late stages of infection, the calculated lifespan of infected cells in vivo, with or without the CD8 cells in the system, was exactly the same. And there was not even a trend towards prolonged lifespan in absence of the CTL. And that experiment was very interesting because, elicited a lot of, it's probably the only paper that I've ever written that there were like 6 or 7 follow-up papers by others, modelists, folks that I've never met in my life. And, I would say about 50% wrote papers to say that this was the worst, piece of crap that they ever seen in their life. And then the other 50% said that was really a fantastic, experiment, creative, and blah, blah, blah. So I think, and it's funny because some of the criticisms were done by people that actually didn't read the discussion, so they attacked us for things that were in the discussion. But point is that it created in my mind this doubt that CD8 T cells can do some strong suppression of virus replication that acts through non-cytolytic mechanisms. So based on that, I decided to do an experiment that was done a few years ago. And this is the only piece of published data that I'm presenting, and I'll go very fast. The experiment was to remove the CD8 T cells in animals that were on antiretroviral therapy for at least 6 months. And I should say at least 2 months, but average 6 months. And the premise of this study was sort of two, there were 2 reasons basically I wanted to do this study. One was to understand whether ART was sufficient, to maintain viremia fully suppressed, when the CD8 cells are not in the system. And also what happened to the virus if virus bounces back after CD8 depletion. What virus bounces back? Is it the virus that was present right before ART, suggesting that there is sort of constant killing of cells that continue to produce virus, or is a virus from the reservoir, suggesting that the CD8 cells are suppressing virus transcription and virus production. So we done the experiment. Was done by Emily Cartwright, a great graduate student in in my lab, who's now a professor at the University of St. Thomas in the most southern state of Canada, which is Minnesota, if I understand. And Emily is doing really well there. And she, what she did, is very simple. She depleted CD8 cells in 13 animals. What you can see here, what you get is a very clear result, that you have animals that are fully suppressed on ART, and those are taking PMPA, FTC, and dolutegravir. And what you get is, as soon as the CD8 cells are depleted, virus production starts becoming detectable. You have positive viremia. And as soon as the CD8 cells come back, viremia goes away. And those are 6 more animals doing all the same thing. And again, this is all published work. But the interesting thing is that when we looked at the virus that was bouncing after CD8 depletion, it was very clear that the virus was not the one that was present right before ART. So we were not seeing the residual sort of virus presence from what was dominating in the plasma, in the replication before antiretroviral therapy, but we were seeing reactivation of early virus that was present at peak viremia and the source inoculum, and that was already replaced in the plasma and in the tissues and organs where virus replication occurs by a virus that has evolved to be CTL resistant and antibody resistant and stuff like that. So this data, again, is all discussed in the paper, strongly suggested that what the CD8 cells are doing in the setting of ART is to suppress in certain CD4 cells, suppress virus production without killing them. These are the conclusion of the study. Again, so the big question in red was, do CD8 T cells silence HIV, SIV expression under ART? So here's where things got interesting. So we decided to do an experiment, and the experiment is very simple. The experiment is, if CD8 T cells do that, one prediction is that if we take a latency reversing agent, an agent that is supposed to reverse latency and induce virus production in cells that are latently infected, and we give this latency reversing agent with or without the CD8 cells in the system, the effect of this LRA would be much bigger when the CD8 cells are not in the system, and it will be lower when the CD8 cells are in the system. Very straightforward experiment. We did it. We took 35 macaques. And as the agent, the LRA, we used Alt803, which now has been renamed N803 because the company Altor has been bought by NantQuest. But let's focus on the 803 part. And the N803 is an IL-15 superagonist. It's a very interesting molecule. You see the design here. It's been used by others. It's a very strong LRA molecule in vitro, but it's not very strong in vivo. In fact, that was one of the reasons we chose it. There was a bit of a disconnect between what it's supposed to do, what it does in vitro, and the fact that it doesn't do the same thing in vivo. So it kind of seemed the ideal candidate to test this hypothesis. And this is the study design. It's 35 animals. 7 animals got the Alt803 alone. 14 animals got the CD8 depletion alone as controls, and then 14 animals got CD8 depletion followed by N803 administration. And in the second part of the experiment, 7 animals of group 2 and 3 received additional doses of N803 at the time of CD8 reconstitution. I will not discuss that part of the experiment. So let's focus on the left side of the slide. So this is just to show, I'm not going to go through this, but we do a lot of RNA-seq. I have a couple of kids in the lab that really love that stuff. So, and what we really, no, it's true, Adam. I mean, I also love it, but they really do. And what happens is that they basically demonstrated that IL-15 superagonist, in fact, works as an IL-15 superagonist. They were able to detect all the gene upregulation that were predicted by IL-15 doing its thing, which is always reassuring, because it tells you that you're doing what you're trying to do. In terms of viremia, this is what happens. So the area that you see in black is the area between the level of detection of our workhorse assay, which is 60 copies per ml of plasma, And the level of detection, the limit of detection of our most sensitive assay, which is between 1 and 3 copies per ml of plasma. So in other words, those animals were suppressed based on the less sensitive assay. They had a little bit of viremia based on the most sensitive assays, but after administration of the IL-15 superagonist had no changes in the basic level of viremia. Very, very clear result. Now I'm going to show you what happened with the Cd8 depletion. So the Cd8 depletion did pretty much what it was supposed to do. Remember that those animals have been treated for 1 year instead of 6 months. So the reservoir is a little bit more stable than in the previous study, the one from the Emily Cartwright paper. And what we saw is that in general, there is a significant increase in virus production after Cd8 depletion. Usually ranging between 60 and a few hundred copies. There are a couple of animals that went around 1,000 and higher. And in general, this is the same effect shown in the previous paper, perhaps a little bit less clear. In the previous paper was 13 out of 13 animals. Here is 11 out of 14, but in the same range. Now I'm going to show you what happened when we depleted the Cd8 cells and treated with N803. And this is the result. So basically, you see that every single animal had a very robust reactivation of virus production, 14 out of 14 animals. A number of animals went above the threshold of 1,000 copies. One thing that is important to note in this experiment, and the way I'm plotting the results, is that every time point that you see in this slide refers to a week after the administration of IL-15. So if you want to, for instance, benchmark this with the LRA study of the TLR7 agonists from Gilead, the GS9620, and I can remember the other one, they were published recently by Whitney and others, and Romas Geratsunas in Science Translational Medicine. They saw all those blips that were lasting between 12 and 48 hours. At the week time point, in their paper, they call it 168 hours, but it's a week, it's the same thing. Every time point is negative. So if the TLR7 paper would be shown with this type of time course, it would be basically a flat line. So this is, to make it very clear, is the highest level of virus reactivation under ART that was ever seen in humans or non-human primates. And this is just some statistics, and I want to show you those 7 animals, because those 7 animals are the most remarkable. So this is the 50% of animals that were treated with Cd8 depletion and N803, and those are animals that had persistent repeated measurements of viremia below detectable limit with the most sensitive assay. So these are animals that have, in essence, less than 1 copy of virus, or 3 copies of virus, depending how much plasma we could collect, for repeated measurements prior to this intervention. And you see how this very full, as full as we can determine with our tools today, suppression of viremia was dramatically reversed and persistently reversed by this treatment. And again, this is a very dramatic piece of work in a sense that clearly the intervention that we have done has changed the way the immune system was dealing with virus production by latently infected cells. You know that I'm not a mouse person, but sometimes, I have to, work with the mouse people. And the reason I've done it, this time is and I'm sorry for the smirk, but, we think that monkeys are a little bit more relevant to humans than mice, but we might be wrong. But one thing we have done is working with my good friend Victor Garcia, University of North Carolina, because Victor and I are both co-investigators in the CAHR Martin Dellini Collaboratorium, and we've been good friends for many years. And And, we always joke, about monkey and mice, and we wrote this review together a couple of years ago. And point is that why don't we stop, arguing, actually do the experiments, in parallel in the same models. And what we found is actually remarkably interesting, because this is the first time, to my knowledge, that an experiment has been repeated, verbatim in monkeys and mice. And we got exactly the same result that you see here. No virus, no reactivation of virus production with N803 alone. Some level of reactivation when you deplete the CD8 cells, and clear, reactivation much higher and most robust in 7 out of 8 mice when they were treated with the two interventions at the same time. And also nice reactivation of virus in tissue. So it was actually very nice and very reassuring to see that in such a different model, the results were confirmed. And it's important because the beauty of the humanized mouse model, and of course I joke, when I make those comments, but the beauty of the humanized mouse model is that you use human cells and the human virus. So confirming those data with the human virus and human cells was really important to us. One thing that I want to make very clear is that this experiment was not an attempt to clear the reservoir. There was nothing here that was going to clear the reservoir. There was no neutralizing antibodies, no CAR T cells, no DARTs, no eCD4, nothing that could conceivably kill the CD4. So we expected the reservoir to be maintained, that we reactivated, but eventually when cells go down, we anticipated that a lot of them would remain as latently infected, when we go back to latency, remain as latently infected cells. So when we interrupted antiretroviral therapy, pretty much all the animals rebounded. So this was not an attempt to reach a functional cure, but rather was an attempt to reach the highest possible level of shocking the reservoir, kicking the reservoir by using this newly characterized or described effect of the CD8 T cells. I just wanted to show you a piece of data that is a separate story, very interesting, just to tell you that what happens with IL-15 is not only happening with IL-15. Here we have another latency reversing agent. It's called AZD5582. It's a SMAC mimetic. This work was done mainly by Ann Cerruti and Victor Garcia at Emory at UNC. Is a non-canonical NF-kappa B pathway agonist, a SMAC mimetic. And what this compound does is activating the virus reservoir and then reversing latency in SIV-infected macaques. In this experiment, you see that 4 out of 12 ART-treated SIV-infected macaques with 12 months of ART reactivated. So there is a real effect. It's a subset of animals that are responding to this drug, and this is in the presence of the CD8. So this suggests that this drug, in a way, acts differently from IL-15 and appears to be more effective as an LRA when the CD8 cells are in the system. However, when we did the same experiment in the CD8 depleted animals, what we observed is that, first of all, 6 out of 6, so we went from 1/3 to 100% of the animals had virus rebounds. And also what we observed is that in this particular experiment, the animals were under 18 months of ART, so even longer ART, and they had also a deepest suppression of virus replication using the most sensitive assay. So with an independent LRA, we also confirmed that the removal of the CD8 enhances the latency reversing agent's property of a given compound. So what's next? Well, what's next is trying to understand what happens in vitro. And that's what we have done in collaboration with Diana Kalpa, who's an assistant professor of pediatrics at Emory and used to be a postdoc for a number of years with Rafik Sekali. And Diana and Rafik developed a system called LARA that is very clever. It's basically you take CD4 cells from HIV-negative donors, you infect them in vitro with 89.6, and then you induce latency through a two-step process. The first is by adding IL-2 and saquinavir, and then adding for about 7 days a combination of IL-7, TGF-beta, and continue antiretroviral administration. So in those cultures, what happened is that you have a peak of virus production, so the cells are GAG positive, and then over time, the number of cells that are GAG positive goes down, but the number of cells that are infected that have integrated virus remain constant. So you go from productive infection to latent infection. It's a very clever, very effective model. And basically, you have a timeline of cells going into latency. And what I'm going to show you is what happened to the level of cells that are still producing GAG at day 6 of this experimental system when you add back CD8 cells from the same donor that have been activated with CD3 and CD28. So this is the level, this is what I just told you about how the system works. So basically, when you sort the CD4 cells, and this experiment was done with sorting the memory CD4 cells and also sorting the central, transitional, and effector memory cells, it's 23 donors. And it's very clear that when you add back the activated CD8 cells autologous, you have a dramatic impact on the level of residual GAG production. In other words, what the CD8 cells are doing here are accelerating the establishment of latency in those CD4 T cells, are working in concert with those latency-promoting cytokines in developing latency. Now, everybody in the room, at least 50% of the people in the room are thinking, oh, but what if these are just CTLs killing the virus? Okay, I'll tell you why these are not CTLs killing the virus. Point number one is because there is no virus killing in the sense that in the experimental system, and I'll show you in the next slide, the level, as you can see on the right, the level of cells with virus is the same over time. So we're not clearing the cells. We're just blocking them from expressing GAG. So it's different. But also, this would be a very, very strange kind of CTL activity because remember, those are donors, healthy donors that have never seen HIV in their life. So for this to be CTL, remember the frequency of CTL precursors in a person that's never seen HIV is like 1 in, a million, maybe, 1 in 10 million. It's extremely rare. So this would be basically either one cell doing all that work, or cells that are non-HIV specific and some mysterious way are doing, what is supposed to be a virus-specific CTL activity. And also, I'll show you in a moment that this is not MHC dependent. So that would be, so there are like several, compelling independent reasons to believe that this is not a CTL activity. And also, we have done class 1 blocking of this experiment, in this experiment, without removing this, this effect. But the first question that I want to clarify, which I think is very important, is that this is not the J-Levy CAF factor that those who are a little bit less diversely young in the room may remember from the 80s and 90s. And the reason is first of all, the CAF was supposed to be present only in cells from HIV-infected individuals, and particularly the elite controllers. But really, what we have seen here is that when you separate the CD4 and the CD8 cells, the effect pretty much goes away. So in our hands, this suppression of virus transcription and this acceleration of latency works only if the cells are in contact. Now we're doing, we actually discussed with Jay this experiment, and he suggested to do, higher concentrations of CD8 cells because sometimes soluble factors may work, and also he's sending us some culture media with high activity of CAF, so we'll see if we're seeing the same thing. But at this point, in our hands is a cell contact-dependent mechanism. And also one thing that is important is that when we use mismatch donors for CD4 and CD8 T cells, we pretty much see the same effect. It's a little bit tiny. It's a tiny bit attenuated, as you can see here. But overall, there is still a decline in virus production and therefore an acceleration in virus latency. So what I told you so far is that there is a strong set of in vivo data suggesting that CD8 T cells induce transcriptional silencing of HIV or SIV in CD4 T cells. And I told you that that is also confirmed in vitro in this experiment. And so far, we have focused on the establishment of latency. And we have demonstrated that this mechanism is cell-to-cell contact dependent, is non-MHC restricted. I can tell you is not dependent on some usual suspect cytokines like TGF or IL-10. But we are getting close to identifying some molecular pathways that could be involved in this, in this process. One important thing that I want to say is that when you look at the activation status of the CD4 cells, and you do it in experiments that are mock experiments, meaning that are the same experiments that you do here, but there is no virus, there's just the CD4 and the CD8, the key effect that the CD8 cells do on the CD4 cells is to tone down their activation. So those are CD8 T cells that reduce the activation of the CD4 T cells. And we go back to that in my last slide. But just hold this thought for a moment. CD8 cells that suppress CD4 activation. So, but the real question in the field is what happened when you try to reverse latency? Because latency is already established during natural infection. What we really want to do, you know, people like Eric and Sabrina and all of us here, what we want to do is reverse latency and then get rid of the reservoir, right? So the system that gives an answer to that is the second part of the LARA experiment, is where the latent infected cells are already induced in vitro. And now you are trying to reactivate them with different latency reversing agents. And this is where Diana and Lavinia in her lab and Julia in my lab and others are working. And basically, those are experiments in which you take cells at the end of the LARA experiment when there is this massive presence of CD4s that are latently infected, and then you add back the CD8 cells. So doing that experiment, what Diana found is, to me, the most striking finding, that this is something that has not been described before. You know, the work is, again, being prepared for publication. We're still doing it with a number of different LRAs. Here I'm showing you the most dramatic experiment using the most powerful LRA that we have, basically mitogens. If you use mitogens on those CD4 cells that have been induced through latency through the LARA assay, you have this very strong reactivation of virus as expected. And that has been demonstrated in many studies. However, when you add back the CD8 cells, again, those are CD8 cells that have never seen the virus before. Those are not CTL. I want to say it one more time. Those are not cytotoxic T lymphocytes. They're not killing. They're not MHC restricted. They come from a negative donor. Those cells, almost at the ratio 5:1, almost completely suppress the ability of mitogens to reactivate virus in latently infected CD4 cells. It's really a dramatic effect. And we're increasing the number, we're increasing the different LRA system. We're now working on sorting what CD8 subsets are doing that, what mechanisms are involved. We have some really interesting ideas. There are some things that they're not ready for being shared because it's only like 2 or 3 observations. But one thing I want to tell you is to keep an eye on the 3 papers, one from John Weary, and that will come up in Nature in about 2 or 3 weeks, describing the lineage differentiation of CD8 T cells, exhausted CD8 T cells, and the role of the TOX transcription factor, because this is all mice work, all LCMV work in mice, but it's really illuminating in explaining how what we call exhausted CD8 T cells are actually far from being exhausted. Our CD8 T cells have taken a different pathway of differentiation that is now becoming very well characterized, and they do a lot of other things, a lot of other functions. But I'll stop it here in terms of what we know about this phenomenon. So in terms of practicality of HIV cure research, why this is so important? Well, if you think about antiviral activity of the CD8 T cells in absence of antiretroviral therapy, probably it's not that important, because whatever the CD8 cells do to suppress virus replication, goes in the same direction. They kill, it's good. They produce chemokines that block the HIV binding to CCR5, like Rantes, MIP1 alpha, etc. That's great too. They suppress virus transcription. That's great. It all goes in the same direction. It almost becomes an academic question, why the elite controllers are elite controllers. Is more the CTL versus is more potentially this effect versus they are particularly good at producing beta chemokines. We still don't know 100%, but in a way it's almost academic. What's really important to think about is that in the setting of curing, trying to get rid of the reservoir, CTL is actually a very good thing because it's supposed to clear an infected cells that reactivated virus. That's the whole premise of the shock and kill, right? So that is very, very positive effect. This transcriptional silencing that we are seeing, and we are reverting in vitro, we're reversing in vivo when we deplete the CD8 cells, and we are inducing in vitro in the experiments that I just showed you. This transcriptional silencing is actually a bad thing that the CD8 cells do in the setting of cure because, in other words, it stabilizes the reservoir. And it makes the reservoir more difficult to clear, at least with the conventional shock and kill approach. And one thing that, again, I want to emphasize is that CTL activity has, of course, no impact on latency reversal per se. It just goes in and mops away the cells that have been activated by something else and have restarted virus production for some other reason. And CTL has no impact that we know on CD4 activation because those are cells that pretty much kill the target cells, and once it's killed, it doesn't matter what the activation is. In contrast, this HIV silencing results in a clear inhibition of latency reversal, but also in a down modulation of CD4 T cell activation. And with this thought, I want to go into the future directions of what we're trying to do. Clearly, we need to, I will start from the bottom, we need to discover the actual mechanism of how CD8 T cells do that. Once we're starting to get there, and once we will be there, we'll be certainly able to pinpoint that and target that, hopefully, which will be much easier in people than depleting the CD8 cells. Although, I'm not pessimistic about potentially doing this in people because we've done much worse things. I mean, they just published a paper, in Nature, a few weeks ago, what's called the London patient, the second, cure patient, which is very exciting, but, it's an intervention that has 25% mortality. Now, of course, if you have leukemia, you have to do it, but what I'm saying, we do Cd8 depletion, rituximab in thousands and thousands of people, and it's not that different than the Cd8 depletion that we're doing, So it's not an impossible thing to think about. But anyway, if we find a mechanism, if we can pinpoint to that, it will be much easier to make that case and to do it. But what I also want to, besides discovering the mechanism, what I also would like to think about are mechanisms of clearing of those cells that have reactivated virus production that are not dependent on the CD8 T cells. And there is a concept that I kind of like, is the concept of, I call it shock and suicide, that is based on the idea of, instead of promoting killing, immune killing of the cells that have reactivated virus, is that promoting apoptosis, cell death of the cells that have reactivated virus. And we now have powerful tools to favor apoptosis. One drug that I'm looking at is a family of drugs are the BCL2 antagonists that have been successfully used in the treatment of chronic lymphocytic leukemia, a disease where there is a deficit of apoptosis in B lymphocytes that become pretty much neoplastic, low-grade neoplastic. It's not an aggressive cancer, but you have this enormous amount of lymphocytes that are circulating, relatively mature lymphocytes that can create problems over time. And pushing them towards apoptosis appears to be an effective treatment. So this shock and suicide treatment is what I am now trying to explore in monkeys. And finally, I want to put one slide. This is my humble slide, because if I ever think that I have discovered anything here, I have to remind myself that, in fact, I've not discovered anything, and that is not surprising at all for those who actually read the literature, that CD8 T cells are able to suppress pathogen replication through non-CTL, non-cytolytic mechanisms. That has been shown for Salmonella, has been shown for Listeria, has been shown for HSV1, has been shown for influenza, has been shown for other pathogens. Unfortunately, the HIV field seems to be sort of hardwired in thinking that CD8 T cells are just CTL, and to me, it's kind of paradigmatic that some people use that as synonyms. It's like nobody would use CD4 and Th17 as synonyms, right? But with CD8, sometimes we do that. And in fact, it's really an oversimplification that creates a lot of unnecessary confusion. The other thing that I've not discovered, and people in the field have known for many years, and there are tons of papers, is that CD8 T cells can suppress CD4 activation. And in fact, the very first description of CD8 T cells by the likes of Stuart Schlossman, Ellis Reinert, Shikao Morimoto in Harvard, in the 80s when I was in high school, was based on that effect. They called them CD8 suppressor cells. They didn't call them CTL, because the first thing that they noticed is when they were co-cultured with the CD4 cells, the CD4 cell activation was going down dramatically. So really what we're looking at here is that somehow, in some circumstances, the CD8 T cells stop acting as CTLs, take a different functional route, probably is a different differentiation pathway, a different transcriptional activation pathway, and they start doing another effect that is, at large, a suppression of CD4 T cell activation, which in the setting of HIV production results as probably an unwanted effect as suppression of virus reactivation, promotion of latency, and persistence of, and favoring the persistence of latency. I think this is a very important point, and again, it's a very different paradigm, and I hope that in the next few months, with new knowledge coming in and the first papers coming out, it will be there will be more people in the field that join us and Victor and Diana and Rafik and the others that are working in this area to try to figure out this mechanism and use it to cure HIV infection. And everything I've done was done by others, as usually happens, at this age. And I just want to show Mirko Paiardini, Anne Cerudi, Wendy Vanderford, Steve Bosinger, Cindy Dardine, Diana Kalpa, and others that really have done most of this work. The tremendous support from the veterinarian staff at Yerkes, they really do amazing things. It's funny, every time I go to them, they tell me that I'm crazy, but then they do it, and then we publish it, and they all get excited, and it's good. People in my lab who did most of the work that I presented are Julia McBrien, Emily Cartwright, Diane Carnatan, Michelle Zanoni, David Palesh, Natasha, and Nikki. Nikki is the one who Nikki now is a professor and vice chair at Miami, is the one who did the experiment that Mario and I discussed many years ago, and has been still involved in this study and has been a very powerful intellectual force behind this line of reasoning. All the money comes from the NIH, the CFAR contributes, the American Foundation for AIDS Research gave us once a grant. My scholarship is from the Georgia Research Alliance of the state of Georgia, and I'm very grateful to Gilead, Merck, and J&J and ViiV for providing us with drugs. And thank you all for your attention.
No, I'll leave it on.